Oh, yeah, I can see that. Okay.
Okay.
Okay, perfect. Thank you so much for your kind introduction. My topic today is past, present, and future of preclinical animal research, reflecting animal research itself. Next. Yeah, animal studies are valuable key components of biomedical investigation, and the proportion of the animal research has increased throughout the year. Yeah, next. Yeah, despite the exponential increase in animal studies, studies are biased and unclear or non-reproducible. So these concerns go beyond the persistent question of how often evidence from animal models is relevant to humans. So the reproducibility of high-profile animal studies seems to be very low, as recently demonstrated in the reproducibility project published in eLife. Next. So Center for Open Science, which has led Brian Nozak, this project showed that evaluated 193 experiments from 53 top papers about cancer published in between 2010 and 2012, and only 50 experiments from 23 papers could be replicated. Moreover, the effect sizes of that fraction were 85% smaller on average than the original finding, and none of the papers had its experimental protocols fully described. And next. So there are many examples of some reported reproducibility concerns in preclinical studies. For example, I remember the very great paper published in Nature Genetics by Professor John Ioannidis. For microarray data, 16 of the 18 studies unable to be reproduced in principle from raw data, and not limited to oncology. In general biology, 80% of 67 studies were not reproduced. And also in genomic cell line, direct comparison of 15 drugs and 471 cell lines from two groups revealed no concordant data. Sometimes commercial antibodies detect wrong antigen, and journal also fails to comply with agreed standards for animal studies or their own statistical guideline. Regarding animal studies, many positive findings, for example, stroke animal studies, there was overt publication bias and only 2% of studies were negative. And in neurological animal studies, which was performed by Silidis and Professor John Ioannidis, they showed that too many significant results over the selective reporting bias. And mouse model of autism, only 10% used correct experimental measures. Next. So this is a Nature survey. 1,500 scientists left the lead on reproducibility. The question was, is there a reproducibility crisis? 52% said yes, significant crisis, and 38% slight crisis, but over 90% said there is a reproducibility crisis. Next. So the next question was, have you failed to reproduce an experiment? The red one is someone else's and pink is my own. Regardless of the field, chemistry, biology, physics, engineering, medicine, earth, and environment, they have failed to reproduce an experiment. The next question was, what factors contribute to irreproducible research? Most frequent thing was selective reporting. The next is pressure to publish, a low statistical power, or not replicate enough in original lab, insufficient oversight or mentoring, and method codes unavailable, and poor experimental designs, and sometimes fraud or insufficient peer review. The next. So in Nature Korea News last year, the title was Publish or Perish Culture Blamed for Reproducibility Crisis. The survey of more than 1,600 biomedical researchers also flagged small sample sizes and cherry-picking data as the leading causes of reproducibility problems. Next. So as you know, many journals had discussed on the reproducibility and waste of research. Lancet, as you know, the research increasing value and reducing waste. And there was challenges in irreproducible research with 12 editorials, and also Science had an editorial and policy forum on the problems with reproducibility. So one biology journal, PLOS Biology, included a section for papers on meta-research in 2016. Next. So research waste had been discussed with Chalmers and Glacios and Professor John Ioannidis, and they in these papers, about 85% of research investment, about 200 billion of investments in 2010 is waste. There are many steps in the research conduct and analysis. For example, in the designing phase, low priority questions or important outcomes not assessed, or 50% of study designed without reference to systematic review of existing evidence. I will show the example later. And adequate to steps to reduce bias not taken in more than 50 studies, and or sometimes inadequate statistical power, or over-reporting or under-reporting with the disappointing result, and more than 30% of trial interventions not sufficiently described, or more than 50% of planned study outcomes not reported. There are many, many causes for the problems in the research. Next. So Professor John, Ioannidis also had a great review on the increasing value and reducing waste in design, research design, conduct, and analysis. In this paper, we can see the in vivo studies. This figure shows the trends in the three methodological quality indicators, like green one is blinded assessment outcome, orange conflicts of interest statements, and blue sky is randomization. It has increased over time, but still very low. The next. So I recognized very great program in Stanford, SPOR, so Standard Program on Research Rigor and Reproducibility, and doing, as you know, you are doing this job, yeah. And next. And also UK, a reproducibility network, and also expanding networks in the world. This is another great thing to improve the reproducibility. And the next. So there is also in the part of journals, reporting standards and availability of data and materials, code and protocols. And it's very great. The Nature journals accept the replication studies. So Nature portfolio appreciate the value of replication of previous findings, and they welcome submission of replication studies that provide new insights into previous published research. Yeah, this is some improvement of from the part of journals. Next. So methodological evaluation of animal studies highlighted the shortcomings long ago, well before the advent of committed reproducibility checks. In 2009, survey led by the NCCRs and NIH Office of Laboratory Animal Welfare randomly selected 271 articles showed poor reproducibility, and researchers are not reported in detail and can't repeat it actually. So in these 271 animal studies, only 59% stated the hypothesis or objective of the study and the animal types. However, 87% did not provide details regarding randomization. 86% did not describe whether investigators were blinded, and 30% did not describe statistical methods. Even high-ranked journal had many statistical and methodological errors. Next. So methodological shortcomings were thought to be happening partly because of a lack of proper reporting guidelines for animal studies. In other fields, the reporting guidelines have been adopted to reduce statistical and methodological biases and hence improving reproducibility when publishing articles. As you know, the CONSORT guideline for randomized controlled trials or PRISMA checklist for the systematic reviews and meta-analysis. So 2010 Animal in Research Reporting in Vivo Experiments Guideline also known as ARRIVE guidelines, were introduced by NC3R, National Committee for 3 Rs. R is, as you know, the reduction, replacement, and refinement. Next. Yeah. So ARRIVE guidelines has been introduced and now adopted in many journals. And so this figure just shows until 2015. At the time, 600 journals adopted ARRIVE, but I suppose that about several thousand paper adopted. But when I checked in some fields in psychiatry and in other fields, they, even though they have a high impact factor, but they did not mention on the ARRIVE guideline. So there is a surge in the support for animal research guideline, but enforcement is a problem. Next. So yeah, this is a Equator network. As you know, this Equator network first meeting was in Oxford in 2006, and it shows the various kinds of guidelines. When I checked yesterday, it shows 699 reporting guidelines. So concert in RCT, strobe, observational studies, Prisma checklist, systematic review, and ARRIVE guideline is adopted here. Next. Also, Equator Network is now adopted in the ICMJE. Next. This is a study, so 2 years later after adopting ARRIVE guidelines, so they evaluate the papers published in PLOS and Nature journals, but they found that there has been very little improvement in reporting standards since 2010. This suggests that authors, referees, and editors generally are ignoring guideline and the editorial endorsement is not to be effectively implemented. Next. This is a randomized control trial, so Icarus trial. So this is an RCT in 2019 to evaluate the compliance with ARRIVE guideline, comparing between either requested completion of an ARRIVE checklist or current standard practice by assessing 762 manuscripts submitted to PLOS ONE. But no manuscript in either group achieved full compliance with the ARRIVE checklist. The details of animal husbandry arrive at subitem 9B was the only subitem to show improvements in reporting. The next. Yeah, so only 9B was slightly increased, but other items did not differ. Yeah. Next. So according to the country, Brazil, United States, China, Germany, Canada, Japan, it shows the similar patterns. So many items are adherence is very low. Next. So our group also performed the adherence of animal guidelines, ARRIVE guidelines, in stem cell studies in animal models of Parkinson's disease. This project also led by Professor John Ioannidis. So we evaluate this adherence to 90 animal studies related to Parkinson's disease. Next. As you can see, the similar patterns with other ones. So some items are adherence is high, but some items are still low. The next. We compared these results between pre-ARRIVE studies and post-ARRIVE studies. Many items look similar, but some items increased significantly. Yeah. Yeah, next. So this was the story of the ARRIVE guideline, but from the part of the United States, ARRIVE guideline is very detailed, but in NIH guideline developed also their own guideline. So this is the very key components. For example, randomization, blinding, sample size estimation, and data handling. These are for minimal reporting requirement, and also they added sex should be considered as a biological variable, for example, in application for preclinical research funding. So this was first proposed by NIH, many departments, but neurology disorder and stroke department initiate this. Next. This is the process the NIH. So they convened major stakeholders in June 2012 to discuss how to improve the methodological reporting of animal studies in grant applications and publications. And this was published in Nature. Next. Also now the NIH guideline shows the review. This is the review guidance for scientific rigor and transparency, and research project grant and mentored career development applications, and they shows the scientific rigor, including these elements: randomization, blinding, and inclusion and exclusion criteria, power analysis, and statistical method, etc. Next. So several researchers evaluate the application of these NIH guidelines to, this is the published in Circulation Research, to cardiovascular disease animal models. Yeah, you can see the various kinds of disease models in cardiovascular field. And stroke is the, regarding randomization, stroke is the highest, but generally the randomization, the reporting was only 22% overall, but stroke is highest. But I think this is due to the many efforts from the, uh, for example, NIH stroke part initiate this process, and also the journal stroke published by American Heart Association. The journal might, the editors and reviewers might recognize this kind of NIH guidelines, so recommend to adopt during revision process or in other processes. So stroke is highest, but others are very low. The next, regarding blinding, the stroke is highest, but over 33%. When compared the therapeutic interventions or not, the most frequently noted in a subset examining therapeutic intervention, 42%, but 28% in others. Next. Regarding sample size estimation, including stroke, the very, very low, only 2.3%. And next. Inclusion of both sexes are only 22%. Yeah. Next. So our group has ever done the methodological rigor in preclinical urology and 14 years systematic review reporting research quality. Next. There are many tables, but this is the summary. So when we evaluated the 3,768 articles, the randomization and husbandry looks significantly increased. The top, the statistical analysis, description of statistics increased significantly, but looks a small increase, but yeah. And regarding blind, it looks similar, but slightly increased. And sample size estimation shows the significant finding, but still the percentage itself is very still low. Inclusion of both sexes or description on inclusion and exclusion criteria at the bottom did not change throughout the year. The next. So this may be due to low awareness of ARRIVE guidelines. So this figure shows various kinds of part academia, the black, and government, pharma, and private institutions, but only very small fraction showed familiar with this kind of guideline and read, heard, but never heard this the most. So low awareness is the first, but another reason is ARRIVE guideline may be superficially endorsed by some journals, may not be mandatory or may not be enforced in practice. Next. So now, 2020, ARRIVE guideline was updated as ARRIVE 2.0. So it prioritized and it described the rationale for each item and detailed explanation and elaborations. So when compared with the original version, so they divided these as arrived essential 10 items, which is a set of 10 minimum requirements for essential to publish to a journal, and optional but highly recommended sets. So blinding is very important part. So blinding has been split from the study design in essential 10 section to encourage blinding and to reduce bias. Also, protocol registration and data access had been newly included in the recommended set. Also, title has been excluded in the version of 2.0 as unnecessary. Next. So this is, if you searched arrive guideline, you can see the original version. This is the similar to the pattern, for example, the strobe or concert title, abstract, introduction, message. There are the guideline items. Next. Yeah, this is the result and discussions. These items should be described. Next. Yeah, this is the Arrive Guideline 2.0. As you can see, the essential items, left and right part is the recommended set. It was divided. Next. So, but it is uncertain whether Arrive 2.0 will substantially improve adoption. So journals' role may be very important, and journals require compliance to Arrive guideline in the introduction for author section. It would be good to have tick a confirmation box of having complied with Arrive for submission completion of animal studies, and should encourage comments about the statistical and methodological transparency of the articles after publications. So reviewers should specifically be asked to be reflect on Arrive Guideline 2.0 when giving feedback, or at least the essential 10 items. Next. So official Arrive Guidelines website tries to promote uptake through specific Arrive action plans for university, journals, and funders. These action plans consist of 3 levels: highlighting Arrive, or implementing the use of the guideline in practice, and monitoring adherence. Next. So this is the study. So whether a change in Nature journal's editorial policy for life science research improve reporting. So intervention of mandatory completion of a checklist during manuscript revision for the Landis for items NIH guideline. They compared 394 Nature publishing groups and 353 matched non-NPG articles described in vivo research. So after mandatory completion of this checklist, increased the description of randomization, blinding, sample size estimation, and reporting exclusion means the data handling. Yeah. Next. But we should think on the reported research and actual research. There is no way to make sure that if an item is checked in a reporting guideline checklist, the provided information is correct. More importantly, reporting will not change the fundamental flaws of studies, of suboptimal standards, or erroneous or biased method. Next. So there is a need for more drastic improvements, and there was also the systematic review, which extracted 58 recommendations on the planning, conduct, and reporting of preclinical animal studies. More than half of the publication recommended same thing: sample size calculation, adequate statistics, concealed and randomization allocation, and blinding. Yeah, same. So multiple efforts at different stages of designing, conducting, and reporting of animal research is likely to be needed. Next. So while further consideration for more improvements is while endorsement arrive 2.0 should be fully supported, much more needs to be done to train better the scientific workforce, to sensitize researchers about biases, to teach them about the best solutions to do credible research, or to change the reward system. So it was my great pleasure to join the students class on the discussing on the reward system. Is very important and valuable to giving credit to the scientists that do the best work with openness and transparency, sharing, and meticulous protection of bias. For genuine improvements, continuous education of researchers is one part, and another thing is the consideration of best practices within the funding schemes. And next. Also, Arrive guideline is on the reporting of animal studies itself, but there is another guideline. The prepare is the a guideline before preparing animal research. This is Norecopa, is from Norway group, so developed this guideline. Okay, so the prepare, so different characteristics. Yeah. So this is the next. This three part. The first part is next slide. First is the formulation of studies. You can check later. So in the various part items. The next. And dialogue between scientists and the animal facility is the second part. Next. And quality control of the components, the study. That is the third part. So this is the prepare study. Yeah. Next. Yeah. So anyway, the most research done in animals is not reported in full detail. Another non-publication issue is the very big problem. So it is not reported at all and remains unpublished. It is unclear whether this happens, but various reasons, including the aims of these projects are not met, or the results are considered uninteresting, or major flaws are discovered, or other reasons. Next. So there was a study, web-based survey among animal researchers on publication practices and incentives for increasing publication rate. This may be by German groups. They found 70% was published among 172 proposals. When they checked the reasons, detailed reasons for no result publications, yeah, one part is the methodological problems, and study was not performed at all, and study was not finalized, or sometimes patent issue. Yeah, next. And they also did a survey and for the suggestions for increasing publication rate. The first thing is the publication cost for open access journals are fully covered by funders or universities, and performance-based allocation of intramural funds for research, reporting of animal study, not supporting the initial hypothesis. If so, they can publish. Also, researchers receive more information from scientific journals that also publish non-significant result. Several journals sometimes receive this kind of non-significant result. Yeah. Next. The value of non-significant research is now acknowledged by many in scientific community because such information can prevent unnecessary animal experiments and offer suggestions for methodological improvement and even lead to alternative hypotheses. Indeed, accepting non-significant research that don't confirm the original hypothesis should be encouraged regardless of the outcome. One way to promote this is to submit manuscript without a research section so that editors and reviewers judge the research simply by its background and methodology. Next. Yeah, this is the study for the publication rate in preclinical research, a plea for pre-registration. This study was led by Netherlands groups. So publication rate, when they checked, the definition was more than one full text or abstract publication, generally over 60%. So it looks similar between small animals and large animals. But when they took a look at the numbers of animals published, yeah, so when they checked, 5,590 animals were used, but when we divided this, small animals described was 23% and large animals described 52%. So they also recommended the pre-registration is important. Yeah. Next. So as you know, for the clinical trials, there is a site for the clinical trials.gov, but in animal studies, there is also preclinical trials.eu. So this group evaluated the 3-year evaluation of a clinical trials.eu reveals room for improvement in pre-registration of animal studies. Not much, yeah, but they show this kind of result. Yeah. Next. So European Heart Journal also recommended, so introduced this site for prospective registration of animal study, but I have not searched for that, but it would be good to evaluate again at this time how many researchers were pre-registered. But I think the awareness is still a problem, and so many, if there is no incentives for like that or in any reasons, so many researchers who are doing animal research might not recognize this. Yeah, next. So also we also invited talk in online the David Moher, very expert in the reporting guidelines. So we discussed on the importance of open science and reporting guidelines in biomedical research. So most of the contents I talked today was the by, collaborative work by Professor John Ioannidis, and he gave many inputs for this paper. So we published 12 years after arrived guidelines. So but animal research has not yet arrived at high standard. Yeah, next. So what should be the future? This is the past, many problems, and we found many reproducibility problems. And so reporting guidelines were introduced, but the adherence rate is still low. So how we can implement and apply the increase the adherence or real in many parts. So I think the reflection is important. So meta research is important for the research go to the right way by reflection of previous research practice. So now we should move forward. There will be many discussions for the future part. The next, yeah. So I think that many evaluation. I introduced several examples for the meta research in animal preclinical animal studies, but there will be many in other sections. So this is just one example. So one trial published in Lancet, the MVA is a tuberculosis vaccine was a trial was done in 2,700 South African infants, but results were negative. So after that trial, several researchers, when they evaluated in preclinical animal studies, except one study, it was found that it was no significant result in animal trial. So uh, as you know, even though the animal study is positive, mostly positive, but if it enters a clinical trial, mostly it fails. But it is failure already in preclinical animal study. So Peter Vivalli pointed out that TB vaccine failure was predictable. So if they perform this kind of systematic review in advance to perform the clinical trial, so we can so we don't know exactly what the result is, but they Vivalli point out this point. The next, I think this is the some kinds of good example of systematic review of animal studies published in eLife. So they Canadian group performed the evaluate the mesenchymal stem cell therapy for sepsis before initiating first in human trials. If they take a look at the animal studies, they also found many problems in the randomization, sample size estimation. They showed that the presence of publication bias resulted in a more than 30% overestimate of the effect, which threat to validity and limit the strength of the conclusion. The result will be similar, but it's important to, yeah, first in human trial should be done, but after evaluating this part, and we can consider the next. The next. So yeah, meta research is important. So as you know, the Nature News 2024 UK now launched the meta science unit. There are many problems in reproducibility, and it means the waste of funding. So yeah, UK might take a look at apply the meta science. So, for example, recently the artificial intelligence many funds will be given to the AI part, but if only AI research is increased, that's also important. But if the funding is allocated to some eccentric to the specific field, some diseases which is very rare, for example, pediatrics, yeah, in diseases might be neglected. So, if meta research can be applied to the funding system in preclinical animal study, we can think what should be the best. So, if meta research is important, I think the funding should be much given to the meta research portion. It's very important. It's like that. The next, yeah, in conclusion, so there have been many concerns on waste and reproducibility issue in animal research. It can be improved by reporting, and the real practices should be changed. And Arrive 2.0 is an opportunity to educate and sensitize the scientific community, including investigators, authors, editors, journals, reviewers, readers, and commentators about animal research, not just reporting, and try to improve also the planning, design, conduct, and the publication of animal research. Such change will require appropriate education, and I think incentives and reward system will be very important. Thank you so much for your attention.